Hello, and welcome back to the Mechanochemistry discussions. This seminar series is hosted by the NSF-funded Center for Mechanical Control of Chemistry, or the CMCC. I'm Ashley Martini. The goal of the seminar series is, as always, to bring the community together. Our seminars stream live on the third Thursday of every month at 10 a.m. Central and are available to watch anytime on our YouTube channel. I encourage you to check out some or all of our previous speakers as part of the seminar series, again, available on the YouTube channel. We also have an excellent slate of speakers lined up for the remainder of this year, and we hope that you will join us to watch them. Before we get started, a great big thanks to a few people. First, Dr. James Batiste, the Center Director, Jennifer Belsick, the Center's Administrative Coordinator, and two students who helped coordinate the seminar series, Quintarius Moore and Noah Sheehan. Thank you so much for joining us. Please do follow us on Twitter as well as YouTube to stay apprised of the activities within the CMCC. Lastly, a few quick guidelines. This seminar is being recorded. If you have questions during the presentation, you can either email them to us at cmccdiscussions@gmail.com, or you can post them in the YouTube channel, and either way, they'll be propagated to the speaker at the end of the presentation. Finally, without further ado, I'd like to introduce today's speaker, Dr. Kristen Hutchins. Dr. Hutchins received her bachelor's in chemistry from the University of Iowa in 2010. She stayed at the University of Iowa for grad school and worked with Professor Leonard McIlvray in the areas of supramolecular chemistry, crystal engineering, and solid-state reactivity, and received her PhD in 2015. Dr. Hutchins went to the University of Illinois at Urbana-Champaign to conduct postdoctoral work with Professor Jeffrey Moore on polymer colloids for self-healing applications. In 2017, she started as an assistant professor at Texas Tech University. Her research group investigates solid-state motion, thermal expansion of solids, improving properties of pharmaceuticals, mechanochemistry, and design of materials that can capture wastewater contaminants. Dr. Hutchins is an editorial and advisory board member for Kristchem.com, and she's an advisory board member for the Women in Supramolecular Chemistry Network, an progression of women in chemistry. Please join me in welcoming Dr. Kristen Hutchins.
All right, so thank you for the introduction, and I would like to extend a thank you to the NSF Center for the invitation to speak in the mechanochemistry discussions today. And so with that, I just want to give the audience a really quick intro into the work that my group has done over the last about 5 years or so. And so our first area, which I'm going to tell you a really short story about today, is our work on thermal expansion properties of organic solids. So essentially controlling how materials, specifically organic materials, respond to temperature changes. The second area, which will be the focus of my talk today, is going to be our work on understanding and improving properties of pharmaceuticals, as well as addressing challenges in the pharmaceutical industry. And then our third research area that we work on is designing materials, primarily polymer-based materials, that will either capture contaminants or valuable resources like rare earth elements for recycling or reuse. And so I'm a chemist by training, and so I kind of wanted to give a brief introduction into sort of the basic type of chemistry that we do. And so when you think of chemistry or organic chemistry, you might typically think of synthetic chemistry, so making new molecules of interest. This is especially true in the pharmaceutical industry, where a lot of efforts are placed into making new drug molecules that will address diseases or sort of provide a cure or a treatment. And so when you think of synthetic chemistry, you typically think of building blocks, let's say A and B, and those are going to come together and connect with each other via a new covalent bond. So maybe a carbon-carbon bond or a carbon-nitrogen bond to link those two components together. And so my group does do covalent bond forming reactions in order to synthesize new molecules that we want to study. But what we probably spend more of our time doing is actually looking at non-covalent chemistry or supramolecular chemistry. And so in this case, we would be interested in looking at sort of the chemistry beyond the molecule. That's what supramolecular chemistry is defined as. And so not just looking at how to make that one molecule specifically, but actually looking a little bit further into how that molecule is going to assemble. And supramolecular chemistry could be done in the liquid phase or solution state, and it can also be examined in the solid state or the crystalline state, where you would look at how that molecule assembles in three-dimensional space. And so in this case, on the top, we have just one molecule, and we might look at how that molecule will interact with other molecules that look just like it. So maybe it's interacting via hydrogen bonding with itself. The other aspect that we could do is we could actually take a second molecule and combine it with our first one and crystallize those together into one solid. And then again, they would be interacting with each other via some sort of force. So what I have shown here in the dashed lines are non-covalent interactions between molecules. So what my group spends a lot of time looking at is hydrogen bonding, as well as halogen bonding, which I'm going to define those for you on the next slide. And some of these interactions might also be things like van der Waals forces, or maybe pi-pi stacking, or even metal coordination. So there's a lot of different tools you can use to actually facilitate those interactions between molecules. And so most people are probably familiar with hydrogen bonding. We teach that in general chemistry. And so in a hydrogen bond, you have a donor atom that is electronegative, and it has a hydrogen that's covalently bonded to it. And that hydrogen is attracted to an acceptor atom. And this acceptor is going to have a lone pair, and it's typically electronegative. So maybe a nitrogen, an oxygen, or a halogen. And so hydrogen bonding is really familiar to most people. It's what's responsible for holding your DNA base pairs together. But halogen bonding is maybe a little bit less familiar. However, it's really similar to hydrogen bonding in that you still have a donor atom, but now in place of the hydrogen, you've got a halogen, so maybe a bromine or an iodine. And this is going to be attracted again to a nucleophilic atom. So it's essentially a pair between an electrophilic region or electron poor region on the halogen with a nucleophilic region on the acceptor. And so that acceptor might be another halogen atom, or it might be something different like an oxygen or a nitrogen. And so these are just two common interactions you're going to see in the talk today. And so I mentioned that we like to look at multi-component solids. And so those multi-component solids that I'm going to talk about today are co-crystals. And so co-crystallization is the process where you combine at least two compounds together to generate a unique solid phase. And so it's not just simply taking two solids and combining them and making a physical mixture of the solids. It's actually that you're going to be crystallizing the two components together to make a solid where both components are present. And usually they're going to be in a repeating pattern. They're typically stoichiometric. And again, they're going to interact with each other by some sort of intermolecular force, which facilitates both of them being present in the solid. And so the reason that I really like co-crystals and think that they're exciting to work on is that a lot of times the co-crystal will actually exhibit properties that are unique when compared to the individual components. And so I want to give you a few examples of work by other groups that have used co-crystallization in order to change solid state properties. And the reason that you're changing the properties is really because you're changing the solid state structure by putting that second molecule into the crystal lattice. And so this top example published by Bill Jones' group is looking at paracetamol, or also known as acetaminophen. And so form one of this molecule is known to have very poor compaction properties. And what they did is they co-crystallized it with a variety of different second components here. And what you can see is the original tablet was sort of easily crumbled, not well formed. But then with the co-crystals, they can actually make really well-defined tablets, so sort of changing that property through the co-crystallization. The second example, published by Tomislav Frisch's group, is looking at dichroic behavior. And so the two molecules on the left are actually single component solids, just one molecule making those up. And the far right is a co-crystal that contains the two molecules on the left. And you can see that the left and the middle molecules have either not dichroic or weakly dichroic behavior, but in the co-crystal, it's very strongly dichroic. So optical properties are another thing that you can tune. And then the last example, published by Christer Ackroyd's group, is looking at modifying properties of explosives. So they chose an explosive that had very poor thermal stability and very high impact sensitivity, so making it pretty unsafe to work with. And then they co-crystallized it, and they actually increased the thermal stability and decreased the impact sensitivity, still retaining the explosive property, but just making it safer to handle. So these are just a few of the properties that you can tune. And today I'm going to focus later on our pharmaceutical work as well. And so when you think about making co-crystals, you can make them via a variety of different ways, but I'm going to walk you through the 2 ways that my group typically makes them. And one of those is by solution methods. And so let's say you have 2 simple molecules here that you might expect to interact by hydrogen bonding. By solution methods, you would dissolve those 2 components in a solvent in which they're both soluble. And there is some tuning that goes into that selection. So you're going to fully dissolve them. You might need to use heat or sonication or something to get them fully dissolved. And then you're going to allow that solution to evaporate slowly over time. And ideally, you will make co-crystals here. And so it might look something like this. This is just one of ours, a vial that we took a picture of in the lab. And so the crystals are a little bit small, but you can see them here, and then a little bit of the solution still left. And so ideally in this process, you will actually get a co-crystal of the 2, meaning both components are present, instead of individual crystals of the 2 components separately. So that's what I mean when I say you have to tune this a little bit, and that sometimes you actually get crystals of molecule A and B independently, rather than getting them both in the same crystal. And so a little bit of tuning goes into that. But then if you have large enough single crystals, you can actually pull one of those out and do single crystal X-ray diffraction to look at the structure. And then you could scoop out sort of the bulk material and do powder X-ray diffraction to confirm that the single crystal you pulled out actually matches this bulk material, and you didn't just pull out one crystal that happened to be good. So powder gives you sort of a bulk idea. You can also do things like spectroscopy, NMR, and IR to confirm that both components are present, and you'll also typically see some shifting of peaks if they're actually co-crystallized. And then you can look at thermal properties as well. And so again, thermal properties are going to tell you a co-crystal versus things like a physical mixture of the 2. The second way, which is going to be a lot of the focus of today's talk, is actually making co-crystals via mechanochemistry or via milling. So mechanochemistry is really simple in that you could actually just use a mortar and pestle, which most people have in their lab, to make co-crystals. So you would just manually grind the 2 components together. In my lab, we actually have a shaker mill. So this is our Formtech Scientific 1000 shaker mill. And so we do this in an automated process instead of by hand. And so in this case, you would take the 2 components, you combine them into your milling jar, and we're using stainless steel milling jars as well as stainless steel milling balls. And there's some flexibility in those materials as well, but that's what we're going to use for the whole talk today. And then you can either do this milling solvent free, where you add no additional solvent, or you might add a few drops of some organic solvent to assist in that milling process. And so then you're going to mill at a certain speed for a certain amount of time. And in the same way, ideally at the end of this, you have a co-crystalline solid here as your product. So you can characterize this. A lot of the times this is going to be a powdered material, so you can pull that out and characterize it by powder X-ray diffraction. And if you happen to have the single crystal structure, you could actually confirm that what you're getting by milling is either the same as what you get in solution, or maybe it's actually different. And then you can do same things like spectroscopy and thermal properties. The only difference is typically you are not going to get single crystals by this method. You're not going to have crystals that are large enough to do single crystal X-ray diffraction. So typically we're working with powder diffraction. And so the first story I want to tell you today is related to our thermal expansion work, and then I will get into the pharmaceutical work. And so this is work by my undergraduate student, Adam, as well as my graduate student, Shouden. And so we had been looking at a variety of different molecules in this project. And this story specifically focuses on using tetrabromobenzene as a hydrogen bond donor and using 4,4-azopyridine as a hydrogen bond acceptor. So we expected that we were going to form CHN hydrogen bonds between these 2 molecules. So a little bit non-classical hydrogen bonding, but still can be effective. And so interestingly, by solution methods, we were actually getting 2 forms crystallizing out concomitantly. And so the first form that we isolated was exactly the form that we expected to get. And so this form is a 1-to-1 form where essentially the 2 components are just held together by CHN hydrogen bonding in an infinite 1-dimensional chain. And so in this case, half of each molecule is crystallographically unique, and the other half is created by symmetry. And so if you look at the tetrabromobenzene down here in the crystal structure, you can see that this half each has sort of 3 different intermolecular forces. One of the bromines is halogen bonding to the azo bridge, the CH is hydrogen bonding to the pyridine, and then the other bromine is halogen bonding to a neighboring bromine. The second form was what we actually did not expect to get. And so in this case, what we have is a 1-to-2 form. So we have half the unique tetrabromobenzene, but now a full unique molecule of the azopyridine. So we do have the CHN hydrogen bond that we initially expected, but now on the other side, we have a halogen bond with the bromine instead of the CHN hydrogen bond. And so again, if you look at that tetrabromobenzene, you can see the bromine to azo bond or non-covalent bond is still there. The CH pyridine is still there, but now this last bromine is halogen bonding with the pyridine instead of halogen bonding with a neighboring bromine like it does on the other side. So 2 unique forms. What's shown here on the screen is sort of a 2-dimensional sheet in how they're interacting with each other. And then that sheet packs via pi stacking interactions with the next one down. And then the pi stacking is also different between the 2. In the 1-to-1 form, we have sort of perfect homogeneous pi stacking where all the tetrabromobenzenes line up and all the pyridines line up. But in the 1-to-2 form, we actually have quite a bit of different pi stacking. We have sort of 2 pyridine rings from opposite ends that line up, pyridine rings that are aligned lining up, and then a pyridine with tetrabromobenzene. And so I won't go into the details of the thermal expansion analysis, but one of the key take homes was that because of that difference in pi stacking arrangements, we actually got 2 times larger thermal expansion values in the 1-to-2 form when compared to the 1-to-1. So a coefficient of 121 versus 286. So even though you have the same molecular components in the co-crystal, because they are arranged differently, you can get differences in properties. And so it might be advantageous to be able to control which form you get if you want a specific property. So then, because we got the same forms in solution and we couldn't just get one of the 2, we decided to turn to mechanochemistry and see if we could actually control the form that we get using milling instead of using solution methods. And my undergraduate William came on to help at this point as well. And so what we decided to do was actually do milling, and we did not add any additional solvent, so just dry milling. And we're using a speed of 900 RPM for a period of an hour, again with our FTS mill. And we chose 900 simply because it's in the middle of the instrument, and it was sort of new for us at the time, so it's sort of middle capability. And we chose an hour because some other folks had been using similar times with kind of similar materials, so that's sort of background as to why we chose those as a starting point. And so I'm going to show you now quite a bit of PXRD data. And so what we have here at the very bottom is the calculated powder pattern for the 1-to-1 form. And then up here in red, what we have is the calculated pattern for the 1-to-2 form. The top 3 patterns are the individual components. So this would help us figure out if we just had physical mixtures. And so azo has 1 form, but the tetrabromobenzene actually has a couple known forms. And then what I have here labeled in black letters are all the experimental data. So what we decided to do was start with a 1-to-1 ratio and then just sort of increase the amount of azopyridine that we were adding in the milling experiment all the way up to a 1-to-4 ratio. And so in the case of the 1-to-1 and the 1-to-1.25, I've sort of highlighted a key peak in the 1-to-1 form. And you can see here, with these lower ratios where they're almost equal, we get almost exclusively the 1-to-1 form. But once we get a little bit above this, the 1-to-1.5, then we start seeing appearance of the 1-to-2 form. And it's not until we get all the way to a 1-to-4 ratio where this peak sort of at about 7 to theta is absent, and we really get exclusively the 1-to-2 form. So we have a little bit more control by mechanochemistry in getting separation of those 2 forms. We do get mixtures in those middle ratios, but then using either 1-to-1 or really strong bias towards the 1-to-2 allows us to get those 2 phases where we couldn't do that in solution. And so now I'm going to move on to our work on pharmaceuticals for the remainder of the talk. And so why we are interested in pharmaceuticals is from a few different aspects. And sort of one thing is when pharmaceutical companies or even academia is developing new drugs, a lot of times there's a lot of target compounds, usually thousands, And ultimately, during clinical trials and development, usually thousands of compounds turn into one FDA-approved drug. And some of the reasons that those drugs fail are maybe poor properties. So things like poor aqueous solubility, poor stability, which would present challenges in trying to give those to patients. Another big issue in pharmaceuticals is the issue of polymorphism, meaning that if you have one molecule, it can actually crystallize into a solid form in multiple different ways. And sometimes those different forms actually give you different properties. So one drug I like to highlight, just because it shows a really excellent example, is the antiretroviral ritonavir, which is used for the treatment of AIDS. And so you can see the structure of ritonavir there, pretty complicated structure, lots of hydrogen bond donors and acceptors. And when ritonavir was being developed, they only were ever able to isolate one polymorph, which is form 1 here. So you can see the crystals are relatively blocky looking. But after ritonavir had been on the market for a period of time, they were synthesizing it in the lab, and actually a second polymorph appeared. And so it's this crystals that are very needle-like instead of these blocky forms. And the difference in those two forms is actually how ritonavir hydrogen bonds with itself. So it hydrogen bonds in two different fashions in the form 1 and form 2. And what was problematic is that form 2 actually exhibited significantly lower aqueous solubility than form 1, which made it problematic when giving it to patients. So sort of in the height of the AIDS crisis, ritonavir had to be pulled from shelves because of this issue, causing a lot of harm to not only patients, but the pharma industry in general. And so, polymorph screening and finding polymorphs and being able to control it is really advantageous. The other aspects we like to address are sometimes there are compounds that really are difficult to get into a solid form or a crystalline form. But when you're talking about dosing to patients, having solid forms to make tablets is really the easiest method. And so addressing crystallization challenges with molecules that just don't like to crystallize is things that we are looking at. And then also mechanochemistry is also relatively green, typically uses no solvent, and could possibly avoid purification steps, which are typically solvent intensive. And so that's another reason we like to look at mechanochemistry. There is a lot of work in using mechanochemistry, specifically in pharmaceuticals. I put some references down here, by no means encompassing, but just a few examples that I think are good and kind of show the power of this. And so this top example, published by Bill Jones' group in 2005, and then talked about in their other paper in 2009, they were looking at caffeine, which all of us are probably pretty familiar with, and crystallizing that with acetic acid. So they did this via grinding methods as well as solution methods. And so when they did a 1-to-1 ratio by grinding, they isolated one form, but a 2-to-1 ratio gave them a second form. And then when they actually did this in solution, where they dissolved caffeine in acetic acid, they were actually able to isolate this second form that they got by their grinding method, but they weren't able to isolate this top form. So I've sort of paraphrased here in this box the takeaway from that, which is similar to a case you're going to see from our lab in just a little bit. In that neat grinding a liquid reactant, so that's the acetic acid as the liquid, they say that that's actually different than just recrystallizing the caffeine in that neat liquid, and that oftentimes you can obtain different products by doing the grinding method with the liquid versus a recrystallization in the liquid. So you'll see this from us in just a few slides. The other example I wanted to highlight by Tomislav Frisch's group is looking at co-crystals of nicotinamide and adipic acid, and they were actually able to find a new co-crystalline form, as well as, which I found really interesting, by changing the milling vessel from PMMA to stainless steel and changing the size of the milling balls, they were actually able to convert between the two forms and control which form they obtain. So this again would be really advantageous if you have a polymorph that is not sort of the ideal form, but possibly you can use mechanochemistry to convert it to the other one. And so this is something we have a little bit of preliminary data on. I won't talk about it today, but we have a system that is sort of giving us two forms, and we can actually use milling to convert between them. And so the first story I want to tell you about with the pharmaceuticals is that of trying to improve properties. And so we chose the drug trimethoprim, which is a BCS class 2 drug, meaning that it has poor aqueous solubility. And we decided to co-crystallize it with nicotinic acid. So nicotinic acid is on the generally recognized as safe list that the FDA puts out. And we typically try to choose compounds that are generally safe so that if we want to put those into pharmaceutical co-crystals and give them to patients, we know that it's non-toxic. And so nicotinic acid was chosen for that reason. Co-crystallization actually gave us two forms. One is a salt hydrate and the other was a salt solvate. The reason these are salt is simply because we have proton transfer from the acid to the base. So it's not neutral. So that's why I've used the word salt. We also used isonicotinic acid, which is just an isomer of the nicotinic acid, so moving the position of the nitrogen on the ring. And in this case, we also got a salt solvate with nitromethane, and all three of these forms exhibited higher solubility than trimethoprim by itself. And so I'm just going to show you the single crystal structures first. And so this is the nicotinic acid with water. And so you can see this is sort of the main interaction between the two components. So this is the trimethoprim, the big molecule here, and then the nicotinic acid. So they interact sort of via a 2-point hydrogen bond. And you can see the acid is deprotonated and the hydrogen now resides on the ring of trimethoprim. So you can see it didn't used to have a hydrogen, but that's where the proton transfer happens. And then the pyridyl nitrogen is actually hydrogen bonding with water, which was crystallizing the lattice as well. In the case of the ethanol solvate, we have a very similar 2-point hydrogen bond, just like in the first case, but now this pyridine is hydrogen bonding to an NH group on the trimethoprim. And we also have ethanol present. It's a little bit hard to see, but I'm just going to kind of circle here as we go. So the OH of the ethanol is hydrogen bonding adjacent to the carboxylic acid group. And so that's sort of where it is at. And then third, we have the isonicotinic acid with the nitromethane solvate. And so again, same 2-point hydrogen bond we've seen before, the pyridine hydrogen bonding with the trimethoprim NH group, which is similar to this one over here on the left. And these sort of make these square-like assemblies. And then we have nitromethane sitting in the cavity of this one. And so we decided to look at the solubility because it was really our goal to improve that aqueous solubility. And so in all three cases, we actually did increase the solubility when compared to trimethoprim by itself. So trimethoprim here at the bottom. And really what we were excited about is that nicotinic acid water salt actually had a 10 times higher aqueous solubility when compared to trimethoprim. The nitromethane solvate did as well. Nitromethane is not safe to give to humans, so this was not nearly as exciting as the case where we have nicotinic acid and water. It's important to look at thermal stability of these systems as well, because you typically modify the thermal behaviors when you do co-crystallization, so things like melting point or decomposition. But it's important to study that so that you don't actually change the stability significantly to the point that you couldn't store it on shelves, for example. And so interestingly, in all of these cases, we saw similar decomposition temperatures when compared to trimethoprim by itself. So really not dramatically changing those properties. And then with the nicotinic acid hydrate, we were also interested in confirming that if we just left this on the bench, we were interested in seeing if it would retain crystallinity or if it would lose water over time. And so we just synthesized it fresh, that's in red, and then set it on the bench for a period of 10 days, just exposed to air. And we could see that it was still incredibly crystalline. Powder X-ray diffraction correlated really nicely to the freshly synthesized sample. So that hydrate has increased solubility, is bench stable. And we did actually, out of curiosity, we're interested in really driving off the water. So we mounted a single crystal on the diffractometer and then heated it up to really force the water to exit. And then at that point, the crystallinity is lost as the water leaves the structure. And it's important to note that the ethanol solvate and the nitromethane actually did lose solvent in air. So when we look at the PXRD data, it does not show as nice correlation when compared to the hydrate system. And so then with those systems, we were interested, so we had made them via solution, and we were interested that since we had some really good properties, if we could make them via mechanochemistry instead. And so again, we're going to do a 1-hour mill at 900 RPM, and we're going to use 1 drop of the solvent that is present in the crystal structure. So water, ethanol, or nitromethane. In the case of a nicotinic acid with water, so the simulated powder pattern is here at the bottom, and we did use a couple different experimental ratios. We didn't do a huge optimization, but we really didn't see excellent correlation to the simulated pattern as much as we would have liked to. So maybe some correlation, but still not getting sort of what I would say is pure phases. On the other hand, in the case of the ethanol solvate, we actually, by milling, got really nice correlation to the simulated pattern. So with the ethanol solvate, we could make that via solution or by mechanochemistry. But with the hydrate, at least not without a little bit more optimization, I would say, with some of these, quick and simple experiments, we weren't seeing really good correlation. And also the nitromethane solvate, we weren't able to get that by mechanochemistry. It probably showed the worst correlation of all three. Okay, so with what I want to address sort of for the rest of the talk is our work in trying to crystallize compounds that present challenges. So compounds that are liquids or oils or natural products are often difficult to crystallize and can suffer from things like oiling out when you try to do a crystallization. And this is work by my graduate student, Daniel, who just defended. The last work was by my graduate student, Chi Xuan, who also just defended his PhD. And so we had been looking at this compound bezafibrate, which is an anti-cholesterol drug, and we had actually made the first ever co-crystals of that drug. And we had went on to try to use some different hydrogen bond acceptors. So we've got this nice carboxylic acid on the bezafibrate, and we were trying to choose some pyridine compounds which could act as acceptors. And so we had chosen the ones I have here on the screen, and these are actually liquid pyridines. And so when Daniel was trying to do co-crystallization in solution, we kept getting oils of these compounds out. And we also would get single components out. And so the reason I decided to kind of stick with this is that bezafibrate is actually a molecule that really likes to crystallize by itself rather than co-crystallizing. And so the beza actually crystallizes in two known polymorphs, and you can actually differentiate those nicely by just looking under an optical microscope. So one is sort of blocky and the other one is sort of needle-like. And so rather than sort of just quitting and saying that this didn't work, we decided, well, there's a lot of molecules that present difficulties with crystallization, especially those that are liquids or oils. And so can we use mechanochemistry to actually get crystallization when solution methods just aren't effective? And we stuck with the bezafibrate because, again, it would be really easy to identify if bezafibrate was crystallizing by itself, because those are so easily identifiable. And that would tell us if if it was working or not. And so what we decided to do was turn to mechanochemical milling. And what we did is we added those two components to our milling jar. So just neat and no additional solvent added. And we milled those for a period of 900 RPM for a period of an hour. And after this milling step, we obtain a viscous oil. And so you might think, well, that's exactly what you're getting from solution. But I was sort of curious and again, pushing it a little bit further. And so I told Daniel, let's take that oil and just dissolve it in an organic solvent and see if we can get crystals. And indeed, we actually were able to get crystals via this pathway. But if we skip the milling and just dissolve the two components in a liquid, we actually did not get co-crystals, or at least I'm going to show you one instance where we did, but not reproducible and not across the board. But this milling step allowed us to get co-crystals via this pathway. So we actually had the first crystalline forms of 3 of the pyridines out of the 4. So the 3 substituted pyridines had never been incorporated into a solid form before, likely because they are difficult to crystallize. So I'm just going to show you quickly the X-ray structures. They are a little bit complicated, but I'm just going to highlight kind of some key points. So with the 2-methylamino-pyridine, you can see sort of a 2-point hydrogen bond there with the carboxylic acid of the bezafibrate. The bezafibrate is sort of partially protonated, partially deprotonated. And the structure with 3 DMAPs, so this one here, is essentially almost isostructural, very similar behavior with the acid groups. In the case of the 3-methylamino-pyridine, we actually got dichloromethane present in the crystal lattice as well. So this is sort of adjacent to these main interactions. And then we had a really interesting case, which mechanochemistry helped us get these 2 forms with 3-PMDA. And so we isolated a 2-to-1 form. So you can see 2 of the drugs here and then 1 of the pyridines. And we isolated a 1-to-1 form. And I'm going to walk you through how we actually found those 2 forms and were able to get one or the other. And so just to summarize this, we initially started with slow evaporation methods. And with all the 3 substituted, we all got oils regardless of the solvent that we chose. In the case of the 2-methyl, just with one specific solvent, we were able to get a co-crystal, but the rest of the times we were getting an oil. So only one specific solvent will give you that co-crystal, whereas oils were very common prior to finding that solvent that worked. And then I do want to note that all of these co-crystals are either fully ionic or at least partially ionic as well. And so the other thing that we wanted to see, since mechanochemistry was working so well, we decided to sort of dig in and try a bunch of different experiments and see if there was anything else that would give us the reproducibility that the mechanochemistry pathway did. And so we also tried slow evaporation in the neat pyridine. So essentially taking bezafibrate and just dissolving it in that neat liquid reactant, kind of similar to that Bill Jones paper I showed you. And in the case of the 2-methyl, we were able to get a co-crystal. In the case of the 3-DMAP, we were able to get this, but we had to use a lot of excess 3-DMAP in order to get this to work. So that would not really be ideal if you, especially if you have precious reagents, to have to use excessive amounts to get it to work versus just stoichiometric. So this does work with a caveat that you have to use excess of the pyridine to get that to work. But in the case of the 3-methyl and 3-PMDA, again, just getting oils out of those experiments. So then we did our neat milling. We actually got oils with 3 of the 4. Interestingly, the 3-DMAP actually did give us a crystalline solid. And so that was a little bit unexpected compared to the rest. And then if we do that neat milling, then slow evaporation, that's where we get these 3 substituted pyridines, giving us co-crystals out really nicely, really reproducibly. And in the 3-PMDA, in this case, we isolated the 2-to-1 form. And I'll show you how we get the 1-to-1 in just a moment. The other experiment we tried to do was fast precipitation in solution. So using really concentrated solutions, dissolving them in an organic solvent and sort of making that crystallization happen rapidly. In the case of the 2-methyl and the 3-DMAP, we were able to get identical materials, but not in the case of the 3-methyl aminopyridine and the 3-PMDA. So we had solids, but just not matching the co-crystalline material. So really at this point, we were kind of curious, so how is milling giving us these results? Because we're doing milling, then slow evaporation, we're putting it back in solution. How is that different than just doing slow evaporation directly? And so we decided to look at some IR spectroscopy. And what you're going to see is actually the co-crystal highlighted in blue, the product that we get from milling in red, and then in green, what you're going to see is a failed solution crystallization. And so by IR spectroscopy, in the case of the 2-methyl aminopyridine, what you see is actually in the IR, you either have signals that are absent or broadened, whereas the oil directly from milling correlates really nicely to the co-crystal. So clearly that product from milling is giving us co-crystalline material, even though it's not necessarily a solid that's easy to work with. By powder X-ray diffraction as well, if you look at the predicted pattern and compare that to the oil directly from milling, you can see that that oil is actually quite crystalline, has really nice diffraction, and shows really good correlation to the simulated pattern as well. In the case of the 3-methyl aminopyridine, that crystallization included dichloromethane. When we look at the IR spectrum, what we see is that surprising for us. And so got us sort of questioning why it's failing when the milling and the co-crystal look really similar. And again, by powder X-ray diffraction, that product shows really nice correlation. But then lastly, what I want to highlight is this sort of odd case with the this new 1-to-1 form. And you can see the IR spectrum of the oil after milling matches incredibly well. And the powder X-ray diffraction also shows really good correlation to that 1-to-1 form. And so how did we actually isolate that form? Because when we first did our milling and our slow evaporation, we got that 2-to-1 form. But clearly we have another form in the mix. And so at that point, we were kind of interested, and now that we had gone through this path of milling and slow evaporation and we were able to get co-crystals, was there any way that we could actually use those co-crystals we had in hand, go back to solution methods and make the material in bulk directly? And so with this case, what we did is we took our liquid pyridine and our solid, we dissolved those in solution, and we took those crystals that we had in hand from the milling and slow evaporation path, and we just added 1 or 2 single crystals to that slow evaporation experiment. And then what we were able to do was actually obtain identical co-crystals via this method that's called seeding. And so we did slow seeding where we allowed that crystallization to happen over a long period of time and did sort of less concentrated solutions and got identical co-crystals in the top 3. And this is actually how we finally isolated that 1-to-1 form of the 3-PMDA. We also did fast seeding where we sort of sped up that crystallization time, used more concentrated solutions and get identical co-crystals. And then interestingly, when we speed up that time, we actually get this mixture of the 2-to-1 and 1-to-1 form. So they are, again, sort of competing in solution. And so we've been able to show that mechanochemistry does facilitate this co-crystal formation. And then once we have those seeds in hand after the milling, we can actually go back to solution and make the material we want in solution methods as well, where we cannot do that without the milling step. And so we were able to nicely have the cover of Crystal Growth and Design in January of this year, which was designed by my graduate student, Daniel. And so in this study, we again showed that milling facilitates the ionic co-crystal formation. And we think that even though it's an oil sort of material, that product must contain particles of the co-crystals, again, as evidenced by spectroscopy and by powder X-ray diffraction. We did decide to let those oils sit undisturbed for several months, 8 to a period of 11 or 12 months, to just see if they would spontaneously grow large enough crystals that we could pull out for single crystal diffraction. But unfortunately, they did not. We could tell by optical imaging that there was, again, these really tiny particles, but nothing that we could pull out and do single crystal diffraction on. And that dissolution following milling gave us the crystals that were suitable for diffraction. So going forward, some things we're interested in doing to really look at this mechanism a little bit more is doing some in situ characterization. So there's a lot of folks who are now doing spectroscopy or diffraction during the milling process, and we think that would be really advantageous for this system specifically. As well as we're looking at if there's a possibility of doing micro ED on the oil directly after milling to see if we can actually see those crystalline particles really sort of at the small level. And I think what was really interesting about this study and kind of shows the power of what the mechanochemistry is doing is that if you take the 3-methylaminopyridine and the bezafibrate and you dissolve those in dichloromethane, again, you get an oil and you don't get the product that you want. But if you go through the milling process and then you dissolve them in dichloromethane, we could get this solvate form. So clearly the milling is offering us a path that solution is just not, and sort of pre-organizing these so that they will co-crystallize together. What we are now doing, it's really preliminary, so I'm not going to show specific structures, but we've now taken some different drug molecules, not bezafibrate, and we are co-crystallizing those with generally regarded as safe compounds, so again on the FDA's list. And we've chosen a system where both of these are actually solids, they're not liquids, but when you do solution crystallization, they just do not co-crystallize. And we've chosen about 4 different drugs that fall into the same class and a couple different GRAS compounds. So solution methods just fail across the board and give us, typically they're giving us crystals of the single components independently rather than a co-crystal. And now what we've seen is by milling across the board, we can actually do milling and have by IR, you can see really nice correlation to the co-crystal. The milling product in this case is actually a co-crystalline solid directly. So we don't have this oil happening in between. We actually make co-crystal immediately after milling. And we now have this high-speed vibrational mixer that we are using as well from FlakTech. And so we can actually do this milling in a period of 5 minutes instead of an hour, much higher RPM. So our throughput is quite a bit faster. We can probably do experiments faster than we can collect data to characterize them at this point. And then you can see here on the bottom, just one example of our predicted powder pattern. Our product from milling shows really good correlation. And we can also take that crystalline powder from milling. We can go back to solution, drop that powder in. So not even like individual crystals, but just the powder directly from milling and seed that material out of solution as well. So there's some wiggle room, but really that milling is providing us pathways to get products when solution methods just aren't effective. And this is work by my undergraduate student Bailey, as well as my new graduate student Mahabube. And so with that, I just want to conclude and say that mechanochemical methods are really useful. We've done a little bit of synthesis as well of just making new molecules, not a ton of that, definitely more in the co-crystal path. And that we're looking at applying these to pharmaceutical co-crystals and salts with improved properties. We're developing methods in targeting compounds that are notoriously difficult to crystallize and trying to use mechanochemistry to control that. And then we are currently expanding to compounds where solution methods are failing and targeting improvements in properties. So with that, I just want to acknowledge my group of students that did the work. So today I highlighted work by Chi Xuan, by Xiaodan, by Mahabube, by Daniel. And then also I had put the undergraduates through there, but most of them have graduated. So this is our most updated picture. I want to acknowledge initially Texas Tech for startup funding when I started the lab. The thermal expansion work that I talked about at the beginning is supported by a career grant through the National Science Foundation and also an ACS Petroleum Research Fund grant. And then all the pharmaceutical work is supported by the Walsh Foundation here in Texas. And with that, I'm happy to answer any questions today. So thanks for your attention.
Thank you, Dr. Hutchins. We have a few questions.
Perfect.
First one, very interesting results. A broad question, why does mechanochemistry lead to different synthetic outcomes? In other words, what is it about mechanical force that leads to products when solution processing doesn't?
I think this is the best question of all. And I'm not sure we have a great answer yet, but I think, you know, as a lot of the audience probably knows, mechanochemistry is a very old technique, right? But it's sort of seeing a reemergence. And I think a lot of people are starting to see this case where we have mechanochemistry facilitating and giving different products than we can access in solution. And so I think there's something different about the mechanical force aspect, which we are obviously lacking in solution, perhaps, you know, increasing molecular collisions, things like this, which is different in the mechanochemistry versus solution. And I don't know if I have a set answer for our data just yet. And I think the in situ experiments would actually give us some light into that. So I'm hoping we can pursue those experiments. But clearly it's giving us a pathway to things that, you know, traditionally maybe you couldn't make, or forms that you would never see in solution. And so I think while I don't have a perfect answer, I just think it's a really powerful technique and really should kind of come side with solution chemistry as a typical synthetic pathway that we look at.
Great, thank you. Next question. Could you please elaborate why fast seeding results in different forms compared to slow seeding in reference to slide 21?
Yeah, so let's see if I can just go back a skosh here. Oh, there we go. Perfect. So in the slow seeding, so again, just to clarify, these are just different concentrations and sort of different speeds of getting those seeding. So slow is taking several days, low concentration. Fast is taking just 1 to 2 days and higher concentration. So we do get these identical forms. And in the 3 PMDA case, which I think might be the most interesting, this was such an odd case where we almost wanted to call it like a disappearing polymorph or a disappearing form. So that a very initial experiment we did with it. So our first milling experiment, first dissolution and solution, gave us the 2-to-1 form. And then following that, when we started doing milling and just repeating our experiments, we actually kept getting the 1-to-1 form. And we did get this 1-to-1 form by seeding. And so I think looking at these two forms, also looking at the structure, I think it's very likely that the 1-to-1 form is the thermodynamically stable form, and the 2-to-1 is probably a kinetic product. Again, especially looking at the structure and sort of thinking about which type of packing or interactions would be favorable. And so since we really had issues isolating that 2-to-1 form, that's why we ended up looking at sort of a faster method, because sometimes if you do things at a faster rate, you can isolate those kinetic products or at least see them. And so that's why this fast seeding, I think, gives us a mixture of forms rather than one, because we're doing it at a faster rate. We're probably seeing that kinetic product because it doesn't have time to maybe convert to the thermodynamic. But then, yeah, identical in these cases. But this PMDA was such a weird case for us.
All right, thank you. Another question. In speed mixing, there can be high frictional heating. How does this compare to running co-crystals in a ball mill?
Yeah, so we've just got the new mill, I think, oh gosh, it's been only a few months or so. So we haven't, the one thing we can do because of that difference is, you know, we ran experiments actually side by side with our old mill. And so we were having to take an hour with our old mill to do the same chemistry. And then in the new mill, we can do it in a period of 3 or even 5 minutes. So much faster, I think, because of that. So really speeding up the time. We can also, you know, the mechanism is a little different, right? It's spinning more like a centrifuge versus the initial mill I showed you is, you know, the side-by-side shaking. And so the speed that we're using in the high speed is also a lot faster. The RPMs are, I think, I think we're starting at around 1500 versus I showed you we were doing 900 in the other case. So we're really increasing the speed. We probably do have a little bit more heat in there comparatively, and we can really decrease the time because of that, because those collisions, those molecular collisions are so much more efficient, I think, in the vibrational mixer versus the shaker mill.
All right, thank you. Another question. Why do you think the crystals are smaller in mechanochemistry versus solution processing?
Yeah, I think essentially that comes down to the fact that because you also have the milling balls present in the mechanochemistry versus solution, you do not have those. So it's really, I think, it's just crushing the material to a finer particle size. And so that's really why you typically don't have big enough single crystals, is because the mechanochemistry is just simply crushing the material. It's also been used as a way to make sort of nanocrystalline materials as well, just literally reducing particle size to nano regime versus these really large particle sizes.
All right, great. Let's do just one more.
Okay.
You mentioned that you used 1 hour milling based on previous work. Have you tested other speeds? And regardless, do you think it's possible to tune this to affect selectivity?
Yes, I do. So we did choose 1 hour based on chatting with the company and a few other folks in the field. And we did change some times as well. We started with some lower times, some sort of 20 minutes, 30 minutes, and not across the board, but in a lot of cases, we were not seeing full conversion to the co-crystal at those lower times. And so we kind of decided that 1 hour with a few of our initial systems was really giving us nice results. And so we stuck with that. I think it's possible, especially maybe in cases where you've got these weird kinetic products or products that form early in the milling, that those shorter times could give you access to those. But yeah, the only problem we were seeing is just still seeing sort of like the two starting materials plus the co-crystal. And so then we increased the time to sort of get the co-crystal reproducibly. But absolutely, I think you could, it might take a certain case to be able to use those shorter times. But yes, definitely tunable.
All right, great. Thank you again for participating in the mechanochemistry discussions. Hello, and welcome back. Thank you again, Professor Hutchins, for that outstanding presentation. And thank you so much for joining us. Just a reminder that we've had an excellent group of speakers so far as part of the CMCC mechanochemistry discussions, and I encourage you to check them all out on YouTube. We also have an excellent slate of speakers lined up for the remainder of this year, and we'll look forward to having you join us as part of the seminar series going forward. Thank you.